Ecotoxicology is a recent branch of toxicology, but has been only partially integrated by environmental policies. This lesson aims to describe the reason to perform ecotoxicological assessment and also the major approaches that can be used to evaluate the chemical contamination of the ecosystem. Until recently, there was no or very low awareness about the effect of potentially contaminated water and soil on the human health, the ecosystem, and also finally on the economy. Contamination problems are very complex. Generally, the reason at the base of this complexity has to be associated to the several exposure sources. And at the end, we have a mixture of chemicals in one or more environmental matrices. Indeed, industry, breeding, and agriculture have a direct impact on the ecosystem, but we have to consider also more unexpected sources of pollution that come from practices associated to daily routine life. To have a precise environmental assessment, several factors have to be considered. First, the actual load of pollutant that reach the environment. Then, the distribution of pollutant among water, soil, and sediment, and no mechanism that may decrease or concentrate them. We have to consider also the transition among different chemical and physical forms as the influence of the environment on this transformation. The pollutants are actually are taken from different organisms, and these have to be carefully considered because it may be the cause of possible transfer along the food chain. An approach based only on chemical analysis is weak and limited. Indeed, analytical methods are not always exhaustive to describe the potential hazard of the matrix of interest. This is mainly due to the fact that few compounds can be detected altogether in an environmental sample, and also because method standards are not optimized for actual environmental sample, because concentrations are often under the detection limit of the analytical method. It's also mostly impossible to perfectly and completely characterize the chemical composition that we have. And so we have to deal at the end with known but also unknown compounds whose danger cannot be predicted. At the end, we have a mixture of compounds that are present and they may work synergistically on the ecosystem, and so we can have unexpected effect on organisms. This problem can be overcome by ecotoxicological information. Indeed, ecotoxicology comes from the integration of ecology and toxicology and evaluate the risk associated with contamination and eventually with the limited analytical data. Water and soil samples are evaluated in their entirety, and so it is possible to study the effect of known and unknown chemicals on organisms, with the final aim to protect the structure but also the function of the ecosystem. The ecotoxicology work with toxicity tests of bioassays, which are based on model organisms. The bioassays are foreseen the incubation of a model organism with the environmental sample, both water or soil, and the test will evaluate specific endpoints. We actually depend on the chosen organism, and these data are compared with control. At the end, a dose response curve allows to give information about the toxicity of the sample. According to the blind dose and the exposure time, tests are used to describe acute or chronic toxicity. As regards to acute toxicity, organisms are subjected to high concentration of potentially polluted sample for a very short time. On the other hand, the chronic bioassays are meant to describe more long-term effect, and so organisms are subjected to low pollutant concentration, but for long and continuous time. The choice of the test to be used is a very critical step. The test should be selected according to several features. First of all, the test should be easy to perform using standard procedures. The cultivation of the microorganisms of the organism should be as easy and cheap as possible, and in order to simplify the protocol, the organism should be small and not pathogenic for animal or plant. The organism should have asexual reproduction and have a fast life cycle. Indeed, should be as much as possible information about its metabolism. Besides, model organisms should be selected among the most sensitive one to the pollutant of interest. Altogether, this kind of information have to be combined with chemical information and also ecological information to give a precise characterization of the pollutant risk in the presence of the ecosystem. As regards the bioassays, they are meant to describe both water or soil sample, but as regards the aquatic toxicity, we have much a higher choice of bioassays. As you can see here, there are several tests that have been standardized to describe aquatic environment, and they are mainly based on bacteria, plant, and crustaceans. As regards soil samples, instead we have less tests that can be used, but we have to consider that we can make liquid extraction from the soil, and so we can obtain a liquid elutriate that actually is representative of the soil sample, and so this elutriate can be used with aquatic bioassays. Here I'm going to present you to show you some tests that can be used among the most common and used tests in the society. First of all, we have the crustaceous Daphnia magna, where organisms are incubated in the presence of 5 milliliters of water, and temperature, nutrient, and light are controlled during the experiment. After 24 or 48 hours, the motility and the mortality of organism is assessed. Another very common test, which is actually very simple and fast, is the bacteria Vibrio fischeri. The organisms are incubated in presence of 10 milliliters of water, and temperature is the only parameter that have to be controlled. Indeed, the test is very fast because only after 15 and 30 minutes the growth of bacteria is analyzed by means of the expressed bioluminescence of the bacterium. As regards the phytotoxicity, we can use several species to analyze the toxicity against plants. We can use both elutriate and water samples. The test foreseen that after 3 days, the germination index is calculated combining the seed germination information with the root elongation ones. Other tests are also meant to describe chronic toxicity, as the earthworm Eisenia fetida. In this case, the organisms are incubated with usually 500 grams of soil. Temperature, light, and humidity are controlled, and the test lasts for also 2 months. After 4 weeks, the mortality and the development of the organism is controlled and is measured, but after other 4 weeks, the effect on the reproduction is analyzed. The springtail Folsomia candida requires less amount of volume, around 15 grams of soil. Temperature, nutrients, and humidity is controlled, and the test analyzes after 4 weeks the mortality and the reproduction of the organisms. Despite the increasing information and increasing knowledge about ecotoxicity, current ecotoxicological risk assessment usually rely with a single species, single test. And this, of course, is not enough because a single organism is not able to predict information that we could have exposed in the whole ecosystem to pollutants. So the use of battery test is recommended, choosing among different model organisms. we should choose among organisms belonging to different phyla and different trophic level. For instance, it should be perfect to select organisms, both unicellular and multicellular organisms, as well as autotrophic and heterotrophic organisms. However, different organisms, different endpoints do not always give coherent results. Each organism has its own sensitivity to the pollutants, and so the response associated to the exposure against a sample is strictly dependent to the specific used organism and the specific analyzed endpoint. So a bacterial test can indeed create a variety of results that have to be merged together, providing a unique response. The ecotoxicological indexes are actually meant for this purpose, so they consider the variability of the test, the severity of the chosen endpoint, and the occurrence of all the acquired data. The obtained toxicity score will indicate whether a sample has to be considered toxic or not. So, ecotoxicological tests, there are a lot of ecotoxicological tests that can be considered standardized, but there is a lot to do in this field of research because pollutants are also changing and the knowledge about their effect is also increasing. So thank you for your attention.